there's a way to make an entrance.
This is my destiny.
It was now a conspiracy of witches.
Download Veely today.
The late Cretaceous, kingdom of the mighty dinosaurs. 100 million years ago, the ancient forests were full of life, not just dinosaurs, but insects, reptiles, amphibians, and tiny mammals that scurried through the undergrowth. The ecosystem of the tropical forest in a prehistoric greenhouse Earth is one of life and death, and sometimes a window into the distant past. In the ancient forests, like today, when a tree is damaged or attacked, it defends itself, secreting a viscous, sticky substance called resin. It protects the tree from infection or infestation. Over the millennia, the forest fell and began to form peat. Then with time, pressure, and heat, this organic material turns to coal. Trapped within the coal is the remnants of tree resin, which turns to copal, then fossilizes into amber. Sometimes things get caught in the resin, organic remains, such as plants of various kinds, even flowers, animals such as insects, spiders, frogs, and lizards. These are called bioinclusions. The amber preserves the creatures entombed within, giving paleontologists a window back in time. Sometimes we find inclusions in the amber that are unique, rare, and exciting. Many researchers around the globe consider amber to be the holy grail in paleontology. And there's a really good reason for saying that, in that specimens have exceptional preservation potential in amber compared to other types of rocks, that preserve fossil remains. So in fact, when we actually look at some of the amber remains, whether they be an animal, plant, or some type of microorganism, they actually look like they died yesterday. So they're in essence in a type of suspended animation over millennia or millions of years. So much so that you can actually see the most finest aspect of their morphology is preserved, from hairs, feathers, on a little dinosaur wing, to all kinds of insect remains and all of their body parts, perfectly preserved just like they died. So as a paleontologist, I need to get as much information as possible from a particular fossil. say you go into a rock quarry and collect fossils, most of those fossils will be from compression because they've been buried for over the millions of years. They've been essentially squashed into 2 dimensions. Amber is very different because those specimens are actually preserved in a 3D space. It's a paleontologist's dream to be able to study these fossils under the microscope and look at the finest details of their morphologies that have been preserved over the millions of years.
So the way we find amber in Australia, and especially in the Anglesey deposit, is a little bit different to how amber can be found around the world. It's very similar to how we find amber or how the Burmese amber is found. That's also found in a coal deposit. But the Baltic amber and the Dominican amber, which are quite well-known amber deposits, aren't found in that way. They're found by eroding that amber out over thousands of years from these deposits somewhere else, say around the Baltic Sea or around the Dominican Republic.
You get some types of resins which are quite clear and golden like this, and maybe a bit darker on this piece here, but some are a red color. You have some that are green. We have Australian amber, which is a very rare green type as well. You get a really rare blue type in Dominican Republic amber. So amber comes in different shapes, sizes, colors, all reflecting the different types of trees, and also what's called the diagenetic processes over the millions of years as well. So you can actually find amber in various ways, mostly from coal, other types of rock samples. You can actually find amber in petroleum wells, of all places. Most of the amber that was found originally in Australia was actually found just on beaches as float. So this was the Cape York Peninsula amber, which turns out not to be from Australia, but from someplace in Asia. And the reason for that is the amber floats in seawater because the density is quite low. But they can travel quite a long ways in seawater with the currents. So it's just surprising the diversity of ways that we can actually find amber in the geological archive.
So the coal comes from Anglesey. It's from the coal mine in Anglesey, and what happened was a bunch of researchers from Monash had found this very amber-rich coal, which we didn't expect to find, and it was very exciting. So they found a few pieces, they took them back to Monash. We've been working on them for a couple of years now, but then the coal mine was about to shut down, so we asked them to get the area where we knew the coal was. We just got diggers to take out a huge bunch of the coal from the side of the cliff, put it in a shipping container and take it here. Of course, this now means that we have a lot of coal.
The coal that we received from the closing down mine, when we went there was a specific horizon where we found a lot of amber, but we just told the guys to get this much. So of all the coal in the shipping container, this much might be the area that has amber, and then within that there's even a smaller percentage that actually has amber in it, even smaller percentage that's large enough to see things, even smaller percentage that's clear enough to see anything in.
So when we first prepare the amber out, or first identify the amber, we'll see it in a block of coal like this, and it'll normally be a small orange or green blob in that coal. And the idea is to try and extract that without damaging it as much as possible. So we use hand tools, little dentistry tools and picks and probes, basically bits of metal with a point on the end, to scratch away at the coal that's surrounding the amber, to try and leave it on a little plinth, a little column that we can then crack off, and then we can take that amber somewhere else.
The issue is the amber is incredibly fragile, so trying to get it out using this method, generally you have to be very, very careful about it, which means that we only really focus on the big pieces of amber because they're the only ones worth getting out this way. Otherwise, we have other methods to get them out.
So in the Anglesey deposit, we get several different kinds of amber. We get oxidized amber, stuff that's been altered by water and the atmosphere as it's been close to the surface. And we also get very clear amber, and we get what we call dark amber. We have cloudy amber of both green and orange colour. So these are these ones here. Unfortunately, that's not very useful for us because it's cloudy, so we can't see into it and see if there's any inclusions. The dark amber is dark because it has a lot of things in it. It's mostly the reason. It has a lot of organic material in it. It has a lot of coal in it most of the time. And then there's the clear material, which is what we want to get out. We want to see clear material that is either green or orange that has inclusions in it. To date, we've found more inclusions in green amber than orange amber. Why that is, we don't really know. The difference in the amber is essentially a colour difference. But we do know that the green stuff has more stuff in it for some reason. We're not sure why. Maybe a tastier tree, maybe more sugar in it, maybe it was more sticky. We don't really know.
A lot of paleontology is a matter of vanishingly small probabilities, essentially. Because if you have an ancient forest, not all of the trees produce resin to begin with, and of those, not all of the resin that's produced is going to contact biology to be included. And then from there, not all forests turn into coal, and not all coal is preserved for us to see. Not all coal that is preserved is found by people or given to researchers or anything. So there's a lot to be explored, essentially.
And eventually we sort through that, get some more clear amber, and that will be amber a bit more like this, which we can start to see into and start to see if there's any inclusions in it. And if we're lucky, we might be able to get a nice big piece out.
Once we have a collection of amber from a piece of coal, we then look at it under a microscope. We paint it with water to clear it up, and we have a look through and see if there's any bioinclusions. So usually we get pretty small bioinclusions because we're working with very small fragments of amber, but there can be bigger pieces, and it is very exciting when we get those bigger pieces because that means we can have bigger bioinclusions.
It can be disheartening to work all the way through a large piece of coal and find absolutely nothing, because then you're just digging through dirt, essentially.
Sometimes we'll find something that's a discernible shape. It's angular, or it's a squiggle, or it's something that could be a mite or a nematode, something very interesting, in which case we sort of separate it out. If it's a big mass of materials, if it's a dark cloud and we can't make out any discernible shapes, then logically there's going to be something in there. We just can't see what it is. So we'll either take it to a high power microscope where we can look through it properly and possibly see any shapes that are coming out from that mass, or we can scan it, which is a very exciting thing to do. You use X-rays to create a 3D model of the amber, and then you can sort of pull apart the shapes that are inside the amber and actually look at what's going on there. Then we can have researchers looking through it and writing papers on the bioinclusions that we find.
We've got to clean all the excess coal off, wash it with a bit of water, but then to see into it, it's sort of like looking into, you know, a stormy ocean kind of thing, like the ripple fractures, and it's not polished, essentially, the amber, so it's difficult to look into without distortions. So to remove the distortions, temporarily we can put it in a fluid like cedarwood oil, which is literally the essential oil I get from the pharmacy. You have to be a bit careful with that. You can't leave it in for too long, and it can mess with the chemistry a bit, but it gives a really good preliminary look into the amber to see if there's any inclusions.
it surprises a lot of people to see when they actually look through a microscope and see the specimen. They can see the hairs on a fly. They can see the legs really perfectly. They can see every aspect of that creature that could be upwards of 100 million years plus old.
And then we look for other fossils within it. So we might look for insects, we might look for plant remains.
If you find something interesting, you can then embed it more permanently in resin, which is little things we make and then polish down.
To protect them, to harden them, to stop them from fragmenting even more, where we then polish that down to be able to see into the amber, see if there's any inclusions in it, and try and identify them from there. In fact, the first time I polished anything was a bit of a disaster. I had a small fly embedded in amber, which we wanted to polish some of the amber back on so that we could get very, very close to it and start to image the eyes. And fly eyes are very complex, so we wanted to try and understand the detail that was in there. And I polished, and I eventually ended up polishing half of the fly away, which was a bit of a mistake on my part. The other half of the fly was there and we could image that, but still it was not a very good result for my very first time trying to polish amber.
There we go. We find a few mites, we find nematodes, we find bits of moss. So these are the really small things that we're talking about, but still they're pretty exciting because this stuff is 40 to 42 million years old. And we don't have a lot of information paleontologically of this period in Gondwana. So any bit of information that we have about any ecology of the region is really, really exciting. We have found little bits of insects, ants. I found something the other day that might be fly legs, and they're very diagnostic of the species, those sorts of things, the antennas and the legs, which is what we find most commonly, but yeah. So my specialty is plants and geology and the interactions between the two things. And those are the things that I really enjoy doing. So this kind of work with amber, which is a plant resin, and finding plants in the plant resin is really exciting. So, yeah.
So amber miners they've been mining amber, which is called bermite there, for various reasons. But the bermite's been mined in northern Myanmar, believe it or not, for over 2,000 years. And this started during the Chinese Han Dynasty. And so over the 2 millennia, miners have been, looking for amber there, which is focused in a fairly small little area of that country. And scientific significance really didn't come to the fore until the last several decades, when we started learning that this actually represents a terrestrial ecosystem that's about 99 million years old. So it gives us a glimpse into that tropical environment. So we're talking about a time when this part of Myanmar was really close to the equator. So we're looking at what's called a Cretaceous greenhouse Earth. So 99 to 100 million years ago. And at that time, heat was being distributed more evenly around the poles, so all the way from the equator all the way to the poles. And it was also a time of increased resin production of trees. So there are various trees that actually produce resin. Okay, and this particular tree is related to the Cupressaceae, which is one of the cypress families. Okay, and these particular trees produce a lot of resin. And once in a while, something absolutely extraordinary will be found. We've actually found a dinosaur wing recently in the Burmite. So a student is actually studying that at the moment. It's quite important because we can actually see the individual feathers of this wing as well as tissue, bones. So we have a lot of work ahead of us to actually try to reconstruct what this wing represents. Is it a non-avian dinosaur or an avian dinosaur? So avian dinosaurs represents early birds, something like that, or is it something else? Is it a type of early theropod dinosaur? So hopefully by studying the wing itself and also the feathers, that we'll be able to find out exactly what that wing belonged to in terms of whether it was an avian or non-avian dinosaur.
So the amber is Cretaceous Burmite from Myanmar. It's roughly 99 million years old. And within this particular piece that I've been studying is a dinosaur wingtip. And so within that you can find feathers, and it can provide a lot of insight into feather morphology and the evolutionary development of feathers over time. Associated with the wingtip is arthropod inclusions, and those can provide us a little bit of insight into ecological interactions. Many theropod dinosaurs, not necessarily just the avians, were believed to have feathers. However, due to the size of the specimen that we're looking at, it's most likely a juvenile. However, small avian dinosaurs have existed within the fossil record. So the difference between feathers within this piece and modern feathers are they are slightly less complicated. The evolutionary history of feathers has seen increasing complexity throughout geological time. And these feathers, whilst they're very similar to those of modern birds, they're not quite there. They lack some of the distinguishing features that would allow us to determine the fact that this is a modern bird as opposed to something that is more primitive. So our next steps in analysing this piece is to be able to try and isolate the different types of feathers that we're finding with it. A single specimen will have more than one type of feather morphology, and so to be able to identify each of those, we can get a better understanding of where this species would fit into the evolutionary history of modern feathers. Through being able to understand past environments, such as that of the 99-million-year-old Burmese forests, it can give us insight in how modern environments would react to things like climate change or other disruptions of evolution of developmental processes. If we can understand how these species and organisms interacted with each other and evolved back there, then it's possible for us to apply that knowledge to current ecology. They have found specimens with quite vibrant coloration, so the coloration patterns that we see in modern birds wasn't necessarily unique to present time. Even 99 million years ago, in the age in which these feathers were first developing, they still had vibrant colours and creative patterns. However, these are rarely preserved within the fossil record, but there have definitely been instances in which we've been able to observe these. But it could definitely mean that a lot of other dinosaurs had vibrant colours that we haven't yet observed in the fossil record, but it could have occurred. So through using the synchrotron scanning that we're hoping to complete soon, it would be possible to look at the bone structure and the bones themselves. Through that, we might be able to identify the species, but also be able to determine the species' age through looking at bone growth signals. And if we can do that, then it's going to give us a lot more insight into what this specimen actually is, whether it's a juvenile or if it's mature, whether it's an already identified species or if it's something that could be completely new.
Paleontology has advanced so much in terms of imaging that there are several techniques that we can actually use to study Okay, and we can actually use various microscopes. We have Leica microscopes we can use, such as the 205C. There's also the BK imaging system that we have here at Monash University. If we want to look internally and externally, we have to use other techniques, such as micro CT scanning as well, and this is a type of tomography that we can use, and also advanced synchrotron radiation as well. So, in fact, we can actually look at this, and we have done so. At the Australian synchrotron, and we did that using what's called the IMBL. This is the imaging medical beam line there. So that has given us information about the internal aspects as well, and also the micro CT scanning.
With this method, I think we just need higher resolution. So the best way is to cut the sample and to do thin sections. So to show you that's looking at the data at 75% full scale. Okay. So this is now the data in full resolution.
People often ask when I'm holding a fossil like this that maybe has some remains preserved, such as a little bit of a dinosaur or something like that in amber, or a feather, can you get DNA out of that? And the answer is no, but we can actually look at the elemental compounds of this too to see if there's anything preserved. And so some ways of doing that are Raman Spectroscopy, FTIR analyses as well, and we've actually found out that there's concentrations of iron, so these would be heme compounds from blood that are still preserved. So you can still get some elemental composition, from this, and so this is advanced chemical techniques that we can actually use.
Raman spectroscopy is a technique to study molecular bonds in our sample. It's based on the interaction of light with matter from the sample. It's more specifically based on inelastic scattering of the light. So the concept is that we flash light on our sample and that light gets scattered, either in an elastic or inelastic way. Actually both. The elastic way means that the scattered energy is the same as the energy of the incident beam. Whereas the inelastic scattering, which is Raman scattering, means the energy of the scattered radiation is equal to the energy of the incident beam plus or minus the energy of the bonds present in our sample. So in other words, through this technique, we can determine what sort of bonds we have in our sample, so what sort of molecules do we have in our sample.
Seeing a spike on a graph. Yes.
Well, I'd love to get a band like that, but...
Yeah. There's still something going on. That's really interesting. In terms of fakes and amber, yes, I've actually looked at lots of fakes just to get a better feel for it. And there's several tests that you can do as well to discern whether the piece you have is actually an actual real piece of amber, or whether it's something that somebody's faked in some way. Because there are specimens out there, and you see them all the time. You go to a market, you'll see a piece that has a big scorpion in it. It looks like it's it's a fake, And how do we test that? And there's various techniques that we can do. We can actually look at the specimens that are associated with a piece like that as well. If there's nothing else in it, usually amber almost always has a lot of organic debris in that too. So that's also an alarm bell that should ring off. If you see a perfect piece, has nothing else in it at all, no other inclusions of any kind, froth or little bubbles, nothing like that, then you could think, oh well, I better check this. So you can actually do what's also called an ultraviolet test, UV test. So real amber fluoresces a brilliant blue, which is quite extraordinary when you look at it. And it's so good, though, that you can actually see the various flow patterns as well, which also tells you that a piece is real or not. When you actually heat it as well, amber starts to melt at around 200 to 250 degrees centigrade as well. In fact, some amber today is used in varnishes and that kind of thing. The black residue is used in varnishes, and it's also used for medicinal purposes as well, in natural medicines and therapies. So you can look at that as well. It actually floats in seawater. So if it just sinks straight away in seawater, you might want to consider that too. And a lot of amber today and little resins are reconstituted, so they've been reheated as well. But what's really interesting that you can see in terms of the inclusions is that sometimes you'll find the finest, what's called stellate hairs or trichomes from plants, which is so delicate that you cannot, if you reheated it, they would be gone. So you can just tell by the association of organic remains. Sometimes you'll say, oh, well, there's a fossil that's only found in the Cretaceous period with dinosaur wing. You say, okay, that, these have been described already. So there are lots and lots of different techniques that we can use, and you really have to look at all of them. But usually a paleontologist who has lots of experience can look at it straight away and say, oh, okay, this is not right, or sure, this looks right to me. Let's do these tests and confirm it. One of the other extraordinary specimens that we found just recently is a section of a little reptile, probably a gecko called Cretaceous gecko. And this particular gecko is so well preserved that the actual skin is there and preserved. We can actually see the little feet as well, but the skin reveals a really complex pattern on it. And you cannot get anything like that preserved in the fossil record unless you look at amber, really, to have that type of preservation in terms of the finest details of the skin pattern. Okay, and it's quite extraordinary. We've also found pieces of Burmite that have snails in them as well. Terrestrial snails are quite rare in the fossil record. We also have invertebrates that we're trying to work out what they are exactly. So paleontologists don't always know exactly what we're dealing with. And sometimes the fossils, because they're 100 million years old, they're groups that became extinct during the Cretaceous period, just before the mass extinction. And they're very different from what's living today. As paleontologists, again, we really want to be able to reconstruct those ancient ecosystems the best we can. We've actually found that so many different types of animals and plants are being found in the Burmite or the amber of Myanmar that it's really giving us major clues as to what kinds of organisms lived in this ancient tropical forest.
So the way we date is to do a process called relative dating. So we have to look at we have to look at the surrounding coal and then where the surrounding coal is in its stratigraphic sequence, where it sits within the Earth, basically. And then we look for other fossils within it. So we might look for insects, we might look for plant remains, but most importantly, we'll look for pollen.
Pollen is incredibly diagnostic of the tree species. So you can look at that pollen and immediately tell, yes, that's the tree that produced that pollen. If we know what period of time that tree lived in, then we can use that pollen to say this amber was most likely produced from this year to this year. And we have found things in the amber that resemble pollen.
The advantage of that is that pollen changes rather rapidly in geologic time. You have species changes reasonably quickly in geologic time, whereas for things like trees and leaves, the morphological changes of trees and leaves are slower.
But if we can use that to narrow down, instead of saying 40 to 42 million years old, we could maybe narrow it down to 40 to 41 million years old. It's still a very broad range of time, but it's still quite diagnostic.
Closer to home here in Australia, we've made some extraordinary discoveries in the Anglesey coal measures. Okay, this was the Alcoa mine, and it really blows our minds as paleontologists because we're actually getting behaviors preserved as well. So we have one instance now, we've just found it just weeks ago, where we have 2 flies that are preserved. So when we started looking at this specimen, we could see one specimen. We thought it was a fly, but we weren't sure. And my eye could just capture something just a little bit deeper inside. So we had to polish it up really well. And lo and behold, we saw one little fly kind of attached to it. And then when we actually rotated it, we could see, wow, they're actually mating. So we actually captured in the resin, which is 42 to 40 million years old. So we're talking about quite ancient times here in Australia. When Australia was still attached to Antarctica, we actually have now a specimen of mating of these flies, which are part of a group called the long-legged flies in the family Dolichopotidae. So it's a really important, and as far as we know, it's the only fossil ever found in Australia that actually shows sexual copulation happening, or mating. So all of our students are creating little captions for newspaper articles and that kind of thing about what they're called, what we could call this, such as doing it Gondwana style or something like that. So there's been a concerted effort in the last few decades to learn more about this Cretaceous ecosystem in Myanmar from the amber, because we do know it's a terrestrial environment, it was tropical. So as paleontologists, we really want to learn more and more about what animals or what the organisms were, what made up this ecosystem in this really interesting part of the world during this hothouse environment. So it's much hotter than today. So CO2 levels were a lot higher, temperatures were a lot higher overall. So it's very different from today in many respects.
It's not likely to impact things that we look at now, modern species extinction, but part of jobs of a biologist and the scientists is to understand where we came from and how we've evolved over time and what our relatives were like.
One of the primary goals of paleontologists is to not only reconstruct the ancient ecosystems through time, which is a really tall order because rocks imperfectly preserve fossil remains. So much so that we think that we only know about a fraction of 1% of all the organisms that have lived through geologic time. So we don't really know that much, but we have to really focus on those deposits that capture all these ancient remains as much as we can in terms of diversity. So we're very fortunate that we have amber, to look at terrestrial environments. As paleontologists, we really want to build a picture through time of the ancient organisms that lived, that tells us more about evolution. As an example of this, in Australia, this is a primary example. Where did ants come from? You ask anybody, so entomologists studying ants, where did they come from? And nobody would know because we've never had one fossil of an ant preserved in Australia. Lo and behold, in the Anglesey coal measures, we have lots of ants preserved. And so they're giving us an insight into ant evolution for the first time, which tells us that groups, and this is the genus Monomorium has lived in Australia and been an important part of the terrestrial ecosystems in Australia for at least 40 million years, when Australia and Antarctica were still attached. But before that, if you didn't have the amber, how are you going to get ants preserved?
It's partly the fascination that we can really see the actual part of the animal that existed 100 million years ago. That's an astounding thing and one of the great privileges of being a scientist and looking at things that nobody's ever seen before. People have been looking for this type of thing for a long time, and it's a great excitement to be able to really see these unique images back into the history of mammals.